Mr. Radford.
Welcome to the Viking Science Academy. We've already talked about mitosis, which is just regular cells making copies or reproducing themselves. But today we're doing sex cells or meiosis. And you say it.
Meiosis.
It's meiosis. I always say meiosis.
Well, no, it's the same. The reason I say meiosis is because, you know, when we talk about single-celled organisms, they reproduce through mitosis. But we go through this process of meiosis.
That's a better way to say it. So in meiosis, we're creating cells that have half the DNA of the original cell. This is important because if a sperm and egg come together, they each need 50% of the DNA. If they have more than that, you can have some messed up offspring or offspring that won't even be born. This process is called oogenesis when it makes an egg, and spermiogenesis when it makes a sperm. And here we have a picture of a human egg being fertilized by sperm. Each of those, the sperm has 50% and the egg has 50%. So what's happening in this? What's different between mitosis and meiosis?
Well, in mitosis, there's exact copies. So we have the parent cell making exact copies to their daughter cells. Okay, in meiosis, the new sex cells are going to be haploid. They're going to have half the number of DNA chromosomes that the parent cell had.
So it's probably going to have to divide more than once.
Yes, it does.
Okay, so it still goes through the same phases: interphase, growing, prophase, metaphase, anaphase, telophase, and cytokinesis. But there's 2 rounds of division. So we have meiosis 1 and meiosis 2. And it's in metaphase and anaphase that we see a difference between mitosis and meiosis. In metaphase, the chromosomes pair up next to each other instead of along a line. And in anaphase, we've got the same chromosomes pulled to the opposite poles. And then later they'll be pulled in half again.
And also, when the second division happens, the chromosomes do not replicate again.
So you can get half.
Yeah.
Now, one of the most important parts of meiosis is something called crossing over. If you have brothers and sisters, why don't all of you look exactly the same? Because you came from the same sperm, the same eggs. Why was there differences? Well, it's because when those chromosomes line up next to each other in metaphase, sometimes the chromosomes kind of swap DNA. You can see from this illustration, then we're going to watch a quick animation on how this occurs. And that means that the 2 sets of DNA that leave and go towards the poles are very much the same, except for some slight differences, some stuff that's swapped. And that's what leads to differences in brothers and sisters. Let's take a look at this real quick. So here we have 2 sets of chromosomes, and when they line up next to each other during metaphase 1, we can see that the tails are going to swap, and they're going to cross over. So we'll see our chromosome on the right swap just a little bit with our chromosome on the left. And when they leave, they have almost exactly the same genetic information, except for a slight little kind of shuffle in it. And it's that shuffle that leads to the wide variety of the human genome. Without crossing over, our DNA would kind of become stale, and we kind of all start to kind of look alike, and everything would seem to be the same.
Yeah, we would not have that genetic variation that we have.
Yeah. So we talked about, there's the phases of meiosis here. We've got meiosis 1, so interphase, cells growing. Prophase.
Chromosomes appear. The spindle fibers start to show up.
Centrioles and metaphase are moving to the poles, and notice how those chromosomes are lined up next to each other, not in a single line. Okay, crossing over occurs, and as they're pulled apart in anaphase, you can see we've got some shuffling of that DNA. So that's the end of meiosis 1, but then it goes right back into the second round of division, meiosis 2.
Yeah. And so again, prophase 2, they do not replicate. Okay, and so in prophase 1 of mitosis and meiosis, that's when they reappear and they start to show up. Well, and they duplicate. In meiosis 2, they do not do that.
We don't want them to. We want to get half of it, so we want them to stay less than half. They're still going to move the equator, be pulled apart, new cells, and now at the end, since we went from 1 to 2 cells in meiosis 1, and now we're going from 2 to
To
4
4.
cells
Yep.
in meiosis 2. And if we look here in metaphase, now they're lined up in a line. They're not lined up stacked with their pair. As they get pulled apart, they now have half the DNA of the original cell, and one cell has made 4 cells during meiosis. This is a big difference between meiosis and mitosis.
Yes.
They make half the DNA. Mitosis, one cell makes 2 exact copies, and meiosis, one cell makes 4 with half the DNA.
Yep.
So we got sperm and eggs.
Yep.
Now, I'm not sure if you knew this, but otherwise they look pretty much exactly the same, right?
Yeah, pretty much.
It's really only our slight differences. So here we have a comparison. We'll be comparing these in class. We'll be kind of mapping them out. It's really important you know the difference between mitosis and meiosis. One thing that is interesting, in men, they can make sperm their entire lives once they hit puberty. So you can be a 100-year-old man and produce sperm and have kids. So I don't know if anyone wants to be a dad and a 100-year-old.
Yeah.
In women, that's not the case. You're only going to produce a certain amount of eggs your whole life, and they're produced when you're born, and then they're dropped. And when those eggs are produced, that one cell created 4, your body decides which one of those 4 eggs is the best egg. And that egg survives, and then your body absorbs the other 3. So it's kind of interesting. Your body's got kind of a weeding out system, and that's one thing that's different with oogenesis than spermiogenesis in terms of how much is produced. Spermiogenesis, they all survive, and males continue to produce sperm their whole life after they go through puberty. All right, but sometimes cells, they forget to stop reproducing, and we call this cancer.
Yeah, it's an uncontrolled self-replication of the cells, and unfortunately, cancer is very
Everywhere.
prevalent. Yeah.
So why is it a problem if a cell just keeps making copies of itself?
Well, I mean, if you think about it, we only have space for a certain amount of cells, and if it starts to just continue to self-replicate, it's going to cause damage to whatever is around it.
It probably is going to draw a lot of your body's energy and resources too, and then it takes over those cells that have a job to do, and they turn into or heart cells or liver cells or lung cells. They stop being them, and they're cancer cells instead.
Cancer cells, and those are called tumors, and it's a bad deal.
Yeah.
Sometimes it's not cancerous. The division can be benign, where you'll have a tumor that grows, but it's not taking over. But
a lot of times not. So to understand cancer and understand sperm and eggs and how those come together and how we have half the DNA, that's why we take a look at meiosis. All right, guys, thanks for joining us on the Viking Science Academy. We'll see you in class.
okay.
Last time I forgot to stop recording.